Okay, good morning, everyone. First of all, this is my first time here in the Regenerative Medicine Conference, and I want to thank the people that accept the possibility that I can be here to show you and to share my modest work with all of you. Yeah? As Professor said, my name is Roberto Ebensberger. I come from the Pontificia Universidad Católica de Chile, yeah, and I want to talk to you now about the nanoencapsulation of mesenchymal stem cells and the possible uses in the regenerative medicine that is join us, all of us here. I like this slide, but I don't want to speak a lot about the many clinical applications and potential uses of the mesenchymal stem cells, all the stem cells already, because all of you know that probably better than I. Yeah. In my lab, we decide to work with mesenchymal stem cells that come from adipose tissue. Yeah, we take the fat from lipoaspirates from voluntary donors, yeah, and then we cultivate or we culture the cells using the classical primary culture enzymatic digestion procedures. Yeah, so here you can see how our cells see in the lab. We have a phenotypification of the cells. Yeah, it's a big one, and obviously, they also differentiate easily into adipocytes, and in this case, also to osteoblasts. Yeah, but once we have the product, that is the cells, what can we do with the cells? Yeah, and I work in a pharmacy department, so delivery is our main objective. Yeah, we wanted to develop novel technologies for the regenerative medicines, and our aim, as the slide said, was the search for new technologies that facilitate the administration, even protection, and or utilization of cells, both in the cellular therapy or in the tissue engineering, yeah? Which this technique also should be scalable for industrial purposes, yeah? And if we also want, also cheap, yeah? So we decide for the cell nanoencapsulation. Now, how will we do this? Yeah. First of all, to all the people that is not involved in the physical phenomena or processes, nanoencapsulation is not all that a nanometric thickness coating layer on the cell surface, yeah, that allows the encapsulation of a single cell, yeah. That's very interesting because the nanoencapsulation, here the term nano is a little bit relative because you work at the micrometrics, yeah, size scale because of the size of the cells. But it is called nanoencapsulation, and the term nano comes here because the thickness of the layer that is deposited over the cell is of a nanometric thickness, yeah, at the nanometric thickness scale, yeah. What happens with this barrier that we create over the cell, yeah, that it can work as an effective barrier against the immunological system, so isolating eventually allogenic cells to trigger an immunological response, for example, yeah. But one of the interesting characteristics that is allowed by the nanoencapsulation that is the free diffusion of the solutes, nutrients, bioactive molecules, and wastes, therefore not affecting the cell viability and functionality. One of the main concerns in nanoencapsulations is that the chemicals that we use should not affect cell viability or functionality. Yeah, that's a main characteristic because we need that the cell has to be functional when they reach the target organ. Now, targeting in drug delivery is a very desired objective, yeah, Dr. Kateb said it in the first lecture on Monday. Yeah, he worked also. She mentioned drug targeting to the brain. Yeah, so targeting is a very, very desirable characteristic in therapy. So cell targeting gives new hopes for stem cell treatment. Why? Because stem cells can move by themselves to injury sites, but not in sufficient quantities, and they do preferentially only to highly irrigated sites in the body. Yeah. Thus, or therefore, the cell targeting search for a cell administration to specific sites in the organism, starting with a non-specific administration. So what we want is to go with the cell in the organism, and that the cell targeting alone can find and concentrate in the damaged tissue, yeah, or damaged organism. The targeting can be done, as said here, by an antibody, but also we can use a specific other protein that is preferentially expressed in a tissue. Nanoencapsulation that we have done is we used not a new method. It's a very known and old method. It's called layer by layer. But the cell targeting here, for example, or nanoencapsulation, is if you put a lot of layers over the cells, yeah, you can immuno-isolate the cells, for example, against the recognition of antibodies. Yeah. Yeah, the layers that protect the cells, yeah, can block the enter or the come of the antibody in order to recognize a specific protein on the cell surface. Yeah. Another thing that you can do for tissue engineering, for example, is to conjugate, yeah, or to adhere extracellular matrix, for example, to the chemicals or the polymers that cover the cells, yeah, in order to the cells specifically attach to a specific tissue, for example, yeah, and the other way that the other thing that you can do is to conjugate an antibody, yeah, and that gives you a tremendous specificity. Yeah, because this antibody, you selected it to recognize a specific antibody, for example, in a cancer or in a specific tissue, for example, heart or the kidney or the liver. Yeah, and you can inject the cells, and the cells will increase their concentrations in that specific tissue. Yeah. Now, how nanoencapsulation is done? First of all, the nanoencapsulation process for cell depends on many factors. For example, the type of the polymer, or in this case, the polyelectrolyte that will be used. Yeah, the molecular mass of this polyelectrolyte, the concentration, the pH, the salt concentration in the solution that you will use, the incubation time. Remember that we are working with living cells, so the incubation times that it's also important, because the cells are a lot of time outside their medium in where they find or they feel comfortable. Yeah. The number of layers, nanoencapsulations allow you to put a large number of layers, one over the cells. Yeah. Much more layers, much more coverage of the cells, and more protected against immunological recognition, for example. Yeah. And the charge of the last layer. Yeah. Why I'm talking about charge? Yeah, because the nanoencapsulation process is a process that is allowed by electrostatic interactions. Yeah, in this case, how it functions, you have the nude cells, but the cells have a negative membrane potential. Yeah, so the first layer of the covering, yeah, that is that we have to select. If you can imagine that this is the cell membrane, you have to choose a polymer or a polyelectrolyte that we will call now polycation because it has a positive charge. Yeah, and you put it in contact here, the cells in a solution that where the polycation is dissolved. Yeah, you allow that both to interact, then you have to centrifuge and wash out all the excess of the polycation in this case. It resuspend the cells. So if you can imagine, here you can see how the cells could be seen after you put the first polymer over the cell surface, yeah? And then obviously you have a positive charge, positive net charge over the cell surface. And then you have to use another polymer that has the contrary charge, in this case a negative charge, yeah? And this case, by electrostatic interactions, the cells, the polyanion in this case, will interact with the polycation and create a bilayer. Yeah? Here, the cells in the nucleus, then in red the polycation, and in blue the polyanion. You centrifuge, wash out the polyanion, and you repeat the steps over and over until you, as many times you want, depending on the layers you want to put over the cells. Yeah, the picture on the right side shows you that every cell is encapsulated individually. Yeah, here the last layer of the polymer has a Fitze molecule that allows the cell to be fluorescent, to fluoresce under the microscopy. So it's a merge image between DAPI and this Fitze. All the cells are individually nanoencapsulated in this case. Another way that you can follow this procedure is by measuring the potential zeta. Yeah? Nude cells, yeah, as I said before, has a negative membrane potential, so the potential zeta is negative. Yeah? But when you put the first layer of the polycation, the net charge over the cell surface changes and turns it into a positive net charge. And the zeta potentials then is now positive. And so on. The next step was to cover the cells with a polyanion, and the net charge over the cell surface goes again into a net negative charge, and the zeta potential goes to a negative potential. Yeah. Sorry. The next step was to see the cell viability of our cells, yeah, after the nanoencapsulation process. Yeah. In this case, we have also used several polymers or polycations in this case, yeah, to look at the cell viability. And excuse me, I forget to put here that our polymer one is in this case chitosan. Yeah. We tried, as I said, 4 polycations, and the only polymer that was useful for nanoencapsulation of our cells in this case was chitosan. As you can say, PAH, that are the name of the polymers are a little bit complicated. PDA-DMAC and PLL, polylysins, are not so effective because they reduce dramatically the cell survival of cells after the process. Yeah. Here is the fluorescence microscopy of all of these bars. Yeah. Here you have the control. All the cells are naturally alive. Then with chitosan, you see that the viability of the cells are preserved and very few dead cells are seen. And with the other polymers, almost all of the cells are dead. Finally, the cell viability using the second layer is the polyanion. We use here a mixture, a 1-in-1 mixture of hyaluronic acid and chondroitin sulfate. And we see that the final process of nanoencapsulation of the cell did not alter the cell viability at all. Salts composition of the medium in where you do the experiments or the nanoencapsulation is also important. Here we used HBSS and Ringer solutions, and both solutions were also enriched in potassium, yeah? Because of the literature I mentioned that potassium was important to nanoencapsulation. But in our case, we demonstrate, or at least in our procedure, potassium was not good. They dramatically reduced by half the cell viability of the process, yeah? So, and when we look at the functional test for the cells, cell proliferation of nanoencapsulated set in black or in green and normal adipose cells are almost identical. That means that the procedure do not alter the functionality of the cells in terms of the capacity to proliferate. When we look at the potential to differentiate nanoencapsulated cells compared to normal cells, or that means not encapsulated, they behave equally, so they can differentiate into adipocytes and also into osteoblasts. What can we do now with the nanoencapsulation? I have to recognize that the first thought that we have in my lab was to increase the mechanical strength, yeah, or resistance of the cells to the extrusion in a spraying device. But I have to recognize also that even the mechanical resistance to the extrusion in a spray is increased by the nanoencapsulation. The cells without nanoencapsulation survive very well the process. Yeah, so there is no difference. Then the other side is what can we do with this nanoencapsulation? And the following idea that comes is to conjugate an antibody, maybe to target the cells. And we look at what happened. And that happens. The upper picture shows the cells that are nanoencapsulated but without the antibody. And the lower picture shows the conjugation of an antibody that was also fits a label. And you can see that the cells fluoresce, meaning that an antibody that has a fits molecule is over the cell surface. In the flow cytometer, you can see also that the cells has a fits molecule in the cells. The cell viability of the process to covalently conjugate the antibody over the surface, yeah, is well tolerated by the cells. Yeah, the procedure of you have to remember that it's a chemical process. Yeah, the cells are submitted to chemical reactions. Yeah. And the viability of the cell is not altered after the complete procedure to conjugate the antibody over the cell surface. And remember that the cells are also nanoencapsulated before. Yeah? We conjugate the antibody to the polymers and not to the cell surface itself. Yeah? Cell viability functionality test show also the same. The complete process of the antibody conjugation do not alter or affect the cell proliferation capacity of the cells. The proliferation curves are almost identical. The differentiation potential is also preserved. Cells can easily differentiate it into adipocytes and into osteoblasts, even when they have conjugated the antibody. And finally, the capacity to recognize a specific tissue. The first experiments we have done with unspecific IgG1 antibody, but here we have conjugated an anti-CD31 antibody over the cell surface of the mesenchymal stem cells when we put it in contact with HUVEC cells. HUVEC cells are cells that express a lot of CD31. CD31 is the PCAM1 protein, yeah? It's an adhesion molecule that is highly expressed over the endothelial cells. And in these pictures, you can see the mesenchymal stem cells alone. They do not have non-nanocapsule, no antibody, and the capacity to adhere to to attract attach the HUVEC cells is known. So in figures here, C and D and E and F, you have the cells only nanoencapsulated, and here are nanoencapsulated only with an unrelated antibody, an IgG1 only, And you can see that the adhesion is very low, and that could be because of the polymers over the surface are only a little sticky, yeah, that recognize unspecifically some targets on the cell surface. But when you put the CD31 antibody that recognizes this protein, the cells attach firmly, and the amount of cells that are attached over the cell surface on the layer of Hubert cells increase more than 100%. Yeah? Okay, that was all what I can show you. And here you can see the Cordillera de los Andes, and this is a scene, a view of my campus in the university in a sunny winter day. Any questions? Yeah, go.
Very nice talk, but I have a question to the cell adhesion, because also integrin receptors are encapsulated, so the cell adhesion is mediated only by sticky capsule or by antibodies, because integrin receptors mediating cell adhesion should be encapsulated in polymer capsule, and the good adhesion is required for proliferation viability. Could you specify more deeply the mechanism of cell adhesion within the capsule?
Good question. I have no answer to your question. I'm not sure if all the integrins are encapsulated and blocked by the nanoencapsulation by the polymers. Yeah. The last figure showed you that the cells alone, without encapsulation, they do not adhere to the cells. Probably there is no interaction between integrin and integrin receptors over the cells or between both cells. Yeah, that can be. And my conclusion through this set of pictures is that the highly attachment of cells on the last figure is exclusively because of the cells have a specific antibody against CD31.
What happens to the capsule as the cells start to proliferate? Because so the surface gets larger, obviously. So the encapsulation should be gone, or at least
it will be diluted with time. Yeah.
Okay.
So over time we have not measured or analyzed what happens, but it's obviously that with the proliferation, also the layers of the polymers will also divide it with the cell. So
yeah, okay.
As the cell divided, the polymers dilute over time also. We also do not know if these polymers goes preferentially to one cells or to one or half by half. It's not known. Okay, but reasonably I have to deduce that the capsule, the layers are diluted on proliferation. Yeah.
So in the end, the protection is gone.
Yeah. Also, the nanoencapsulation is for the first time. You have all the single cells encapsulated, and you can put it into the systemic circulation, and then the cell and find the target, and then can divide and forget about what it has.
Okay, thanks.
Can you evaluate the time stability of this capsule?
Excuse me.
How long these encapsulated cells are stable and do not lose capsule in PBS or in serum?
Excuse me, I didn't understand. My English, sorry.
So cells are encapsulated, and these capsules, how long this capsule is stable under the cell surface?
We have not measured it.
Capsule in the PBS solution or in serum, because a lot of protein can bleach.
Yeah. Now I understand. We have not measured how many times the cells are viable over time, but in other kind of experiment that we have done in the lab, I have to say that the viability of the cell in suspension, if you mean from when the time I make the process until I send it to the clinic, yeah, it depends on the buffer or the solution in what you have the cells. That's the main important. It's not the nanocapsule. The nanocapsule do not affect the cell viability. So,
No, not viability, stability of the capsule.
Stability.
In certain solutions, is the capsule diminished? Is it unstable in some solution? Is that what you're asking?
Yeah.
Well, good question. I do not answer. But I think that are electrostatic interactions, so they are very strong. So I find very difficult that they live alone in solution. Doctor.
Great talk. I have a comment about the nanoparticles. Are they capable of conjugating and basically creating a microparticle?
Grain?
Are they capable of conjugating and creating microparticles? Because one of the major problems with nanoparticles or nanopolymers is that conjugation, and that conjugation can be toxic to kidney and liver.
Yeah.
So biodegradability is important, but have you looked into conjugation?
I have to say that these are not nanoparticles. These are micrometric size. The size of the final particle is the size of the cell.
Right.
Only the thickness of the layer is at a nanometric scale. Yeah, it is not a nanoparticle.
So the nano he's using as the measurement of the thickness of the layer rather than...
We can avoid the nano prefix, and we can call single cell encapsulation. Yeah.
Since the assembly is actually pure electrostatic, there's a lot of other proteins. Usually, even in vitro, there's a lot of other protein. When you grow it in serum, like 10% in a capsule, and some of the protein got charges themselves.
Mm-hmm.
So they're probably gonna inside your structure. And you haven't shown any proof that it's actually pure encapsulation of chitosan. So, everything's showing that it's encapsulating around, very pure and so on. I haven't seen any proof of that. Could it be that it's sort of a chimeric, a lot of protein, chitosan inside it, just attached to the cell, and there's no real encapsulation?
No, we have done measure. Also, the interaction of the polymers are purely electrostatic.
Yeah, but there's other proteins which also bind electrostatic.
Yeah.
Except depend on the PI, right? Isoelectric point.
Mm-hmm.
So everything can bind, like albumin, even everything. So how you know that your encapsulation is a pure chitosan?
No, we have done measure it.
Okay, that's something that's